All right, it's my pleasure to introduce Dr. Noor-ul-Huda. She's a postdoctoral associate supported through the postdoctoral scholarship program of St. George's University, True Blue in Grenada, in cooperation with Mississippi State University's College of Veterinary Medicine. She earned a bachelor's degree and 2 master's degrees from universities in Pakistan and a PhD from the Chinese Academy of Sciences in Wuhan, China. She's currently stationed, in what I understand, the last 6 months or so, at the Thad Cochran National Warm Water Aquaculture Center in Stoneville, Mississippi, working with Dr. Matt Griffin and Dr. Dave Morancic on a variety of aquatic animal health topics, and is going to present today on proliferative gill disease in the U.S. catfish aquaculture industry. So you can take it away.
Well, I hope I'm visible as well as audible to all of you. And thank you so much, Dr. Nick, for the introduction. You really did great with pronunciation. And I'm also thankful to American Fisheries Society for giving me this opportunity, as well as to all of you guys for taking out time for my talk today. With some of you guys, I may have met in Eastern Fish Health Workshop, but for some of you, I may be a new face. So as introduced, I am Noor-ul-Huda, and I have background in fish nutrition, and that too in dietary probiotics, gut microbiology and metabolomic side. And I recently joined Matt Griffin's lab as postdoc associate to work on a very interesting research project related to PGD. I'll talk about that one too. So in today's seminar, I'll be sharing some information related to PGD, its status, past and present, especially with its role on mitigation strategies. And with that, I have my notes and water bottle with me. And I'll be sharing some major takeaways from the recent studies. And when I say recent study, that means 10 years or so, because Mississippi PGD has been a problem for catfish industry since last 3 decades or so, and there is a lot more information already present. So I'll try to take give you some basic insights. Along with that, there are tireless efforts being made from scientists all over the world, especially scientists at Mississippi State University, who are tirelessly, putting their efforts to bring some good results against PGD in catfish. So I may take a little more time today, longer than usual. And those who have attended Eastern Fish Health Workshop this year, they know that Matt could not finish his presentation in time somehow. So you guys have missed a lot of information. So here I am, Matt's postdoc, presenting what he could not present, along with effect of fingerling size on proliferative gill disease severity in channeled catfish, which is a very highly corroborative study between among USDA, MSU, and the University of Georgia. So stay tuned with me for that. Well, I like this graph from USDA sharing some statistics related to US catfish industry and the status. Like it says that catfish farmers in US have produced around 343 million pounds of food fish per decade in last years or so. And Mississippi states accounts for more than 54% of the total fish production. And that too is mainly because the farmers have adopted improved production technologies as well as farm management practices. But this industry at the same time is facing a lot more challenges. There are more, but I'm showing only main 4 of those challenges. They are like rising in feed costs because ingredients, like good quality ingredients, are getting expensive day by day, or farm management is getting costly as well. Moreover, industry is also facing competition from imported fish, which is relatively cheaper compared to the locally grown fish. And last but not the least, the infectious diseases. And we all know that they not only affect fish health, cause mortality, but they put additional financial cost onto the farmers. With that, here is a notorious disease, which is the 3rd most diagnosed infectious disease and the most prevalent parasitic disease of channel and hybrid catfish, proliferative gill disease. It's another name is hamburger gill disease because of the appearance, like meaty appearance it gives through the gills. The information now we know about it is that its etiological agent is Hanagia telleri, which is a myxozoon, and it has a typical myxozoon life cycle where it has an intermediate host. In our case, it's channel catfish, where it undergoes myxospore stage, and it also has a final host, which is an invertebrate, Dino digitata, where it undergoes actinospore stage. And fishes, catfish of all sizes are susceptible to PGD, but small size fish or juveniles are more, severely attacked. The symptoms include the respiratory distress because the gills are in constant exposure with the actinospores. They give the swollen gill lamellae, as we can see here too. Another typical symptom is the chondrolysis, where we see cartilage breaks in gills. And I see these days, farmers are bringing their fish, and the Lester Koo, he takes out the gills and make the wet mounts. And the first thing, sometimes he asks me that, what is the first symptom? Then I say that, oh yeah, it's a cartilage break. So I'm learning. Moreover, we have multifocal interlamellar hyperplasia of the branchial epithelium. And well, as we human beings, we get cranky when we are sick. So fish does too, and they become anorexic or they stop eating. So the information that I have presented in these two slides, it was not gathered over a period of 2 years or so. There was a constant efforts of a decade in the past, mainly because of, I lost my surprises. Anyways, mainly because of these names who really did not need any introduction. Let me share a story. When I joined Matt's lab, he gave me papers related to PGD, as well as certain theses, like big theses, because he wanted me to, understand what is happening and what has happened in the past. And there was a big chunk of information for me to digest and process. But I couldn't help imagining a fairy tale where Catfish is an innocent princess and Hanagayas are the villains, because there are, I think now 11 Hanagayas that cause they use Catfish as a host. And these guys are the superheroes or the legends who are constantly trying to face the innocent catfish against PGD. Moreover, they have a huge role in detecting H. in pond water too. I'll talk about it soon. Well, in the past, there is a paper I would like those who are interested, I would like them to read this paper, because this was possible, detecting H. in pond water. Before that, they were using fish in the cages, or, they were using sentinel fish. So that has its own history thanks to the David Wise. And yeah, I'll move on, but you guys can check out this paper. This slide I got from Matt's presentation. Well, I did add a couple of more pictures here, and he calls them the next generation. In future, I'll be talking about the major contribution by Graham and Justin. And for me, their work comes under the heading of, genetic control of the PGD, because they really have worked a lot on myxozoon diversity and metagenomic studies. We have Ethan Woodyard with us, who is able to find, animals and birds vector who are contributing in PGD. This is some very interesting piece of information coming soon. We also have Fernando Yamamoto, who is leading the nutrition lab at NWAC. He has background in nutrition. I have background in nutrition. So together we met with Matt Griffin and my co-advisor, David Marancic from St. George's University. We are working on nutritional aspects, or, to having some feed additives and trying to break the life cycle of H. telluri, which is a part of our postdoc project. And I hope we'll be able to bring some good results in future for you guys. So stay tuned. Talking about preventive measures and mitigation strategies, one thing is for sure, it is kind of a well-established fact, thanks to the efforts made in the past, that no chemotherapeutic agents or other curative treatments occur or exist against PGD till now. all of these genetic, the controls, call it biological, genetic, chemical, or certain management practices, have their own benefits to a little extent and their own limitations too. I'll check out all of them one by one, just a little bit, so that you guys can have the idea that none of these strategies are 100% effective against PGD. That is why we are seeing a lot more cases each year after another. Talking about the chemical control, here is a big list of the chemicals that have been used to mitigate PGD, either including in the diet or treating water, pond water with chemicals, or using certain chemicals to control the population of Dira digitata, which is the definitive host. But the limitation for this chemical control strategy is that it is cost prohibitive. The chemicals are really costly, and also a lot more quantity is needed so that they could reach to the bottom, and which is not good for fish health in a long run. And also we can add additives in the diet that we are working on. They are somehow effective, but there are a lot more questions lying ahead of us in this area. Talking about the management practices, as I mentioned that fish undergo respiratory distress, and for that we can install paddle wheel aerators, which is cost prohibitive. They need money. But there is another way that we can transfer the fish from the infected pond to a clean pond, right? But we know that fish do not go without gills anywhere. So if the fish go with the gills, the parasite also goes to the fresh pond as well. So that pond would not be clean anymore. Another strategy which is being used since a decade or so is to plan for stocking the catfish ahead of, in the spring season when it's the right time, so we can properly plan when to stock fish in the ponds. And that is the second part of my presentation too. So the third one is the biological control. Well, here they used fathead minnows, smallmouth buffaloes, or another mosquito fish, thinking that they would eat the echinospores in the pond water. But you know what happened? Well, oh sorry, we are online, so I can tell you what happened. Catfish started eating these fish species. And these fish, because they have gills as well, so they started acting as a reservoir for echinospores too, contributing towards more diversity of myxospores. But somehow not much luck. But I would like you to refer this paper by Richardson. It is a nice paper where he used 5 species, and so the major takeaway from this paper is that he was able to find less copies of Metictylari 18S ribosomal RNA when they challenged the fish co-stocked with mosquito fish. If I'm not wrong, I need to make sure. Yeah, so they used that one, but the results were quite good after 24 hours post-challenge. When they took the samples after 7 days, they were not so much good. So that means still there is a lot more work to do in future on this front too. I'll talk about the genetic control, where hybrids are produced crossing male bluefish and channel female catfish. And thanks to the efforts of Matt Griffin, now we know that parasites have lower transmission rates of this disease. That was the case in the pond. So they came up with the idea to challenge the fish in tanks. So the methodology they adopted is like they took water from the ponds where they already have an active PGD outbreak. They transferred it in the hauler to this facility at NVAC, where they stocked hybrids and channels. So one of the studies we get from Roser, I'll just mention the major takeaway. He was able to find arrested development of H. in hybrids as compared to the channels 14 weeks after this challenge study. And the main observations were a large number of mature parasites were found in channels, but hybrids didn't show that much. And Rother was also able to bag some of the novel myxozoan spores of genus Senegaya, which is a good thing. But this study had its limitation that they were not able to find parasites from tissue sections. So they planned another study. This time they came up with a better plan, which was to make molecular probes targeting signature DNA sequences in the tissue. That solved the problem thanks to Stilwell. He used in situ hybridization. Here you can see the image. So he successfully found spores in channel catfish 14 to 20 weeks post-challenge here, but he was not able to find anything in the hybrid fish, which gave rise to the idea that hybrids are the dead-end host because they were challenged at the same time in the same facility. So, but we are seeing stuff in channel catfish, but not in the hybrids. So what's going on? That was the question, right? So hybrids are the dead-end host, or what happens after 20 weeks or so? Where do these spores go? These questions are still unanswered. Moving on to, but before moving on, when they found the age thing from Justin's work, they thought that it's kind of a big deal, which is nice. But why do we still see PGD in hybrids? Why do farmers are still losing the fish? What is happening? So that was kind of, unanswered question. Well, moving on, we have another study. They did, which is a continuous production plan, another better plan where they have, we still have rather, I'll call it. We have at NVAC, these 18 1-acre ponds. So they stocked channel catfish in 8 ponds, 9 ponds, and then hybrids in the rest of the other ponds. And they took water samples every month or so to check the metagenomic studies, right? So here are the results. You can see here, they are the plots showing results from 2015, 16, and 17. And here is channels. These graphs are showing a community structure there, myxozoan community structure, and here is a hybrid. I would like you guys to focus on this. For me, it's like a pink or mauve color, but yeah, you can take it as whatever you like. So here's a pink and the blue. I would like to focus on this one. The pink or mauve is showing the Echectolari, whereas mauve is showing, the blue is showing Henegaya axillus, which is another myxozoan species. So we can see more diversity or more population of Echectolari in channels compared to the hybrids, which are somehow preferring H. axillus. And I can't stop but think like what would be happening in the ponds. Maybe, channels must be talking to H. axillus that, oh, not in my pond, please. You're not here, or you're not allowed, or maybe they have certain preferences. These are some questions lying ahead of us too. We do not know, in simple words. With that, we have more work and more questions. And all of you who are listening or who want to join us or contribute in this ongoing projects, you are welcome. Sorry, madam, I'm speaking on behalf of you, but yeah. So we have this work like using non-ectolerate fish as decoys, and what is the feasibility of crop rotation, because if there is a dynamic, community dynamics is kind of different in ponds stocked with channels or in ponds stocked with hybrids. So what if we stock them in the same pond one by one and see what they're doing with, myxozoan community? Maybe they will suppress it. We do not know. And also, as Ethan did, role of birds or animals as vectors, how they are contributing. Why do some ponds have more PGD than others? Are hybrids really created equally? Are they really the dead-end host? What if we add antiparasitic feed additives? Will we be able to break the life cycle of Echectolari? More questions. We need more support and more funds, of course. But here's the good news for other folks. Related to one of the important mitigation strategy that we are following at NVAC since last 15 years or so, but with a little background here. So in this graph, you can see proportion of fish gill damage compared with relating with the water temperature, which rises across a period of a year. So we see a peak of PGD when the temperature is quite high in April or May or June, kind of we call it a PGD season. And farmers lose a lot of fish, almost like 100% or so as well. So that makes spring season a crucial time period to plan their stocking wisely. So farmers have these concerns in springtime, like they want to know when to stock fish for the grow out ponds, or if they had bad luck with PGD in the past, they do want to know when they can restock fish in the ponds. So for farmers in Mississippi Delta, we have this molecular testing facility at NVAC for the pond water, where we perform ejectillary specific qPCR to estimate ejectillary levels in the pond water. That remember I mentioned using the sentinel fish by David White, and then leading towards the idea of checking ejectillary in pond water. So farmers bring water sample and we check them. Actually, Cindy Bear, she checks them, and results are usually given in the same day, but if not the same day, then the very next day. So here is a sneak peek of how the conversation goes. So a farmer comes with certain questions or concerns like, I want to stock fish in the ponds, but I suffered with PGD loss last year, or I lost fish due to PGD, when can I restock, right? These are some genuine concerns. So here comes Matt Griffin, who says that, okay, bring water samples of your pond and I'll check them. So the farmer, he brings water samples, and usually they are not in this much quantity. They are like 10 or so samples in a day. And Cindy Ware is the lady who single-handedly, because I'm witnessing it here, so she does all the processing. She spins the water bottles, she extracts the DNA, she runs the qPCR. So hats off to her, along with other responsibilities that she has in the lab. So she does that, and based on the results, Matt suggests, like, okay, you can start stocking in the low risk ponds, but here comes the red flag. Some of your ponds are still hot with ejectillary, and he does not suggest them to restock in them or stock in them until they are in a safer zone. And I was told that at times ponds remained hot for a good 6 weeks or so, and they kept bringing water, they kept checking them, and they were not in a position to stock until. But farmers, they do not wait, because it's a matter of money as well. So they keep asking, when can I restock? Or some of them came up with the question that, what if I stock a larger fish, because PGD is more severe in small size fish? Well, to check this question, 2 trials were conducted actually. So in trial 1, 40, 80, and 120 pounds per 1,000 fish were stocked, like 3 different sizes of fish were stocked in the ponds which had PGD. And the number of ponds were 8. We used like 10 fish per cage, and they were exposed in this water for good 7 days. And this study was conducted even before I arrived, but I was fortunate enough to perform some, qPCR analysis. Hence, I am here. So in trial 2, just to increase the number of observation, we used only 2 sizes, extreme sizes, small and the big one. And this time we used more number of ponds. From 8, it increased to 11 with 10 fish per cage and the same duration. So the parameters assessed were survival of the fish. Gill clips were taken for wet non-exam for chondrolysis. Gill arches were taken for histology, and that was this part was done by Dr. Alchemis from University of Georgia. And gill clips for qPCR estimate of parasite burden were taken, performed at NVAC, whereas pond water was also taken for qPCR assessment of actinospore level at NVAC. So Dr. Brad Richardson, Bradley, he's the guy who from USDA. So he did all statistical analysis on the results of these 2 trials using R and GLM. He did perform analysis on medium-sized fish too, but the results were somehow inconsistent and making complications. So we decided on showing results or conducting analysis on only two sizes, small and the big one, for the two trials together. So in next few slides, I'll be showing the results only of these two trials together. So yeah, we were able to find some chondrolysis. You can follow the arrow here. The cartilage break was there in the gills. Whereas histopathology that was performed by Dr. Alkimis at University of Georgia. Since I'm not a pathologist, but I'll try my level best to do the justice with this finding. So here is a normal gill. For those who are new like me, it's for you guys. So they say this is a normal gill where we can see a centrally located filament and literally extended, gill lamellae. And here is a gill which is showing a severe case of PGD. And these cases or these kind of gills were found in most of our fish in our study. So we can see extensive loss of gill filaments. We don't have it here. We got expansion of filaments by intense granulomator inflammatory cell infiltrates. Epithelial hyperplasia and hemorrhage was also seen. And whereas these dots, these are the pre-sporogonic plasmodia within the areas of inflammation. PGD lesions were also counted, and for that, actually, 10 of the longest sequential filaments were taken from 3 gill arches, from 3 gill arches per fish, and they were counted for the lesions that include cartilage break, inflammatory cell infiltrates, hemorrhage, as well as epithelial proliferation and number of pre-sporogonic plasmodia. So here in this picture A, B, C, and D, we can see the inflammation where here is this part is showing severe hemorrhage. This is showing cartilage break, and this big guy is a regenerative chondrodysplasia photo. So this infection was quite severe in both sizes of the fish. Here are some results from the pond water qPCR analysis. So we did check pond water CQ against number of lesions per fish. And for, again, those who do not know, here CQ is indicating the values of parasitic DNA. So if the CQ is higher, that means low parasitic DNA is present. And if CQ is low, which is indicating parasitic DNA in the pond water was quite high. So here we can see the mean number of lesions or the lesions in the gills of the fish, they were quite high when more parasite DNA was present in the pond water. Irrespective of the fish size, we did not find any statistical differences in both sizes of the fish. Same was the case with the pond water CQ against mean pre-sporogonic plasmodia and the gill damage. So we found that as the pond water CQ increases or the parasitic DNA decreases, PGD severity also decreases with no marked difference between fish sizes. So that was the case with the pond water. Let's look at the fish gill qPCR. So this plot is showing fish gill CQ values of parasitic DNA here and mean number of lesions. The case is kind of same. We see a high number of parasitic copies here causing more lesions in the fish or more disease, irrespective of the fish size. Kind of same results were obtained or observed with when we compare the values of fish gill CQ with mean number of pre-sporogonic plasmodia and the gill damage. So that led us to conclude that as the gill CQ increases, that is showing less parasitic DNA, PGD severity also decreases with no marked difference between fish sizes. So were we just lucky enough to have these consistent results or so? Before you think anything else, here is I promise this is the last graph. So here is the survival analysis. And I would like to say thanks again to Brad for adding the term survival here. I asked him to do so because I do not like the term mortality, is quite negative. So thank you for putting it. But here we can see proportional survival against pond actinospore concentration. So the criteria is same, high CQ, low DNA, low actinospore concentration. So I would like you to focus here where we can see less survival or more mortality in the fish with more actinospore concentration, and then situation gets better in both sizes of the fish when the actinospore concentration is kind of low. This observation is somehow validates the risk assessment model being used at NVAC for the catfish farmers since last 15 years or so. And this is the key that Matt uses for the results. Here, we say that if the CQ is lower than 28, the pond is said to be on a high risk, so you can't stock fish in there. Or if you stock, you will lose it, of course. Or CQ between 28 to 32, if the ponds have these kind of CQ, they are considered to be in moderate risk, and they prefer to, hold them for future until they are in a low risk or a better zone, which is the CQ higher than 32. So although that was not the plan of conducting these two trials to validate this risk assessment model, but somehow all the results, be it like fish gill damage or lesions or survival, they are contributing towards strengthening the idea of using this model or keep using it in future. And on a very important note, I asked Matt that since how long we are doing here, and he told me that we started with doing 100 ponds a year, and then when the results were fruitful and the farmers somehow they were getting some money and some encouraging results. Now the number of ponds that we check at NVAC has increased from 100 to 500. That means more work, and we do not charge anything to the farmers in this front too. So coming back to the questions of the farmer that stocking larger fish would save him against PGD. Well, from these two trials and the results, we say that we do not find any benefits from stocking the larger fish during PGD season. We are not economists, and I won't go into the debate of stocking larger fish or the small fish because they may have their own benefits. It's their personal choice. But based on these two results, well, PGD does not discriminate based on size, gender, or the sex of the fish. This is the first take-home message. And the second take-home message is that it is not the size of the catfish which is a predictor of when to stock fish during PGD season. Rather, it is the actinospore level in the pond water that is the main predictor, or that decides when to stock fish in the pond. So the last take-home message is, if the pond is hot, please do not stock your fish at all. This is the last slide, I'm sure. So yeah, those who are interested, this is the risk assessment model, the official, I forgot the word. The official poster, we have it at NVAC, and I would like you guys to look at here, where the criteria is if the spores are more than 50 per liter, well, that is a risky zone. And some real-time photos from what Cindy does or what we do in our lab. These are the water samples, all running at one time, spinning them down to a lesser quantity where we can extract the DNA, and then we do the qPCR and do the results. And that's how Delta region or NVAC is helping farmers, catfish farmers in the community. That is the end of my presentation. I would like to acknowledge all of you guys for your patience, and all of these guys here who somehow contributed towards these studies, and a lot more names I could not add here, but yeah, thank you everyone for working against PGD. We are in this together, and the show must go on. And I would like to say thanks to the Catfish producers for allowing access for this particular project. With that, I would like to acknowledge my affiliations of PSPS, GU, MSU, CVM, MAFIS, DRAC, or USDA. I hope with this presentation, the newcomers may have found some information useful for them, and the experts also may have found something, interesting on updates related to mitigation strategies. So thank you so much, everyone, for your time. And I'm available for your questions.
Great. Thank you very much. That was an incredible presentation. It's great to see the amount of work that has been done on this disease and the value that it's brought for the industry. So congrats to all of you down there. We've got plenty of time for questions. So if you want to drop them in the chat, or feel free to raise your hand or unmute yourself and ask the question directly.
I have a question. Excellent talk, Noor. Good job. I really enjoyed it, and I cannot believe that Matt was planning to cover all this in 15 minutes, in addition to what he talked about already in the Eastern Fish Health. But a quick question. You said PGD is ranked number 3 or the 3rd infectious disease in catfish. What are the top 2?
They are bacterial diseases, actually.
Which ones?
Thank you for testing my knowledge. I need to work more on that. Of course.
Columnaris, Aeromonas, ESC. You have to draw one of these, right?
Columnares and EC, and which is the third one you mentioned?
Columnaris, Aeromonas, ESC.
Columnares. Lester, please do not listen what I'm saying.
Anyways, I just wanted to check with you to see if I'm up to date or not.
Yeah, thank you so much for your inputs.
Excellent talk. Thank you.
Thank you.
Just for clarification, it's columnaris and ESC are number 1 and number 2.
Thanks, Matt.
May I ask a question?
Yeah, sure.
Great talk. Thank you for sharing all of your hard work. I had a question about the hybrid versus channel susceptibility. And from what I could tell, but maybe I missed it, you guys were always measuring parasite loads in the gill tissue, right, by PCR. And then my question is whether you think that there's some sort of difference in susceptibility to infection at the earlier stages before the parasite makes it to the gill. I'm assuming, like other myxozoans, this has like a blood stage. I don't know much about the biology of this specific myxozoan, but if it has like a blood stage like the others, do you think that there's differences in how much of that gets in the blood prior to getting into the gills?
Well, that is an amazing question. Since I'm new to PGD, well, I do not find myself a fit person for this, and I would like to defer this question to Matt, actually, because he's the right person to answer this question. I'm sorry. Matt, please.
Hey, Irene, good to see you. So we're not sure where it goes. So some work that Graham did where we actually followed parasite DNA throughout the whole body, like in all the tissues. Within about 4 weeks post-challenge, we can't detect it. And then around between 12 and 20 weeks post-challenge, it returns to the gills in the channel catfish, but in the hybrids, it does not. So everything we've done so far, we've been able to produce myxospores in the channels, but we haven't been able to do the same in hybrids. We're not sure if they're just a full-on dead-end host or if we're just not waiting long enough to look. But yeah, that's on the radar. We're with Justin's in situ hybridization now, we have more tools to be able to actually follow the progression of the parasite through the fish, and so that's certainly something that's on cue. But yeah, I don't have a real good answer for you other than we don't know where it goes, but we know it leaves and it never comes back in the hybrids.
Yeah, it's super interesting. Thank you. Yeah, with the in situ, you will be able to, at least on blood smears and things like that, maybe try to see if there's any any differences there, although we might have a completely different biology because these guys are so complicated, these type of parasites. But yeah, it was very exciting work. Thank you for sharing everything.
Great. Any other questions out there? All right, if not, thank you very much, Noor, for your presentation today. We really appreciate it. It was great.
Thank you so much for having me here. And thanks to all of you guys for listening.